Yes, I'm my own technical moderator, which is very convenient. Um, okay, so welcome everyone. I want to talk to you about my master's project. I'm going to show you the results that we have after 3 months of work. Um, this is my project, Construction and Analysis of the Moniliophthora roreri pangenome. So yes, I come from University of Sao Paulo. So first of all, chocolate is in danger. Moniliophthora roreri is a fungus that actually attacks cacao cultivars all over the tropical strip of the world. And as you know, chocolate comes from cacao. So I really like to use this to kind of capture your attention. I hope I did. Basically, as I said, it's mainly in the tropical side of the world. Up to date, we have a lot of outbreaks of Moniliophthora rorei in cultivars of cacao in South and Central America, mainly. Um, the fungus actually entered Brazil just a few years ago, 2018. We had the first outbreak in Brazil. Um, it mainly attacks species of Theobroma, which is cacao, and we have also Errania. And it's actually now moving on to another kinds of hosts like cupuaçu, which is another Amazonic fruit. So yeah, we also have a very closely related fungus, which is Moniliophthora perniciosa, that causes witch's broom, which is a very similar infection. So we know this fungus has a hemibiotrophic life cycle. However, we don't really know the pathogenic mechanisms of the fungus. We're still not sure about the biological or the geographical, sorry, origin of the fungus. We don't have a lot of information about the reproduction of style of the fungus. So as you can see, there's a lot of gaps around this pathogen, and we're talking about a pathogen that destroys almost 80% of Colombians' cacao a year, for example. So yeah, we kind of said, okay, we need to save cacao from this fungus, and we need to kind of understand a little bit better who is the enemy so we can actually attack the enemy, right? So for that, we decided to build a pangenome, which we already had a really nice introduction to. But yeah, we mainly have a pangenome where we can separate the accessory genome, the core genome, and the exclusive genome of a species. And we can have pangenomes built orthology based, or we can have graph based pangenome. And in this project, we're actually trying to make a comparison between the two methods using a previously built orthology based pangenome that was also made by me in my grad thesis. So for this project, we're working with a data set that was published in 2023 by Andrea Minio et al. At UC Davis. This is actually the most complete set of genomes that we have up to date of Moniliophthora roreri. As you can see, there's not a lot of genomes for this fungus, so it's really hard to get a lot of information from it. We have 5 of those samples that were built with PacBio and Hi-C, and then we have short reads with Illumina, that it's the rest of them. So for this first exploratory part of the project, we are actually working only with the 5 most complete genomes in order to kind of standardize the tools and the pipeline so we can then scale it to the whole data set. So this is just a brief method section. So we have the genomes, we assemble the graph-based pangenomes, we classify the segments, annotate, do a little bit of enrichment analysis with Go terms. We did a variant analysis, and then we compared everything. As you can see, this is a lot of stuff. So I'm gonna go just really briefly through everything. So on the right, you can see your ortholog-based pangenome, and we see the pangenome classes. as I said, we have hard core and soft core. Those are like the core, the main genes that a species needs to maintain itself, to survive. And then we have accessory and exclusive. And this was really interesting when we built the graph-based pangenome, because we found in the accessory and exclusive the majority of the genes, which is not very usual, because you would expect you would have more genes in the core, right? In the essential genes. So that was one of our most interesting findings at first. So then we said, okay, let's see, what do we have inside those genes, those genomes? So for this, we made a Go term analysis, Go enrichment analysis, and we actually managed to find a lot of enriched Go terms. We decided to cluster them in these 5 clusters you're seeing on the right. Um, there, so once again, we found, as we expected, the Go terms that are enriched in the core class in the core pangenome are mainly related to survival, to development, to tissue growth. However, when we went to look at the accessory and the exclusive classes, we found some very interesting Go terms related to viral genome replication, for example. We found antibiotic biosynthetic processes. We found a ceramide metabolic process, which is reported in the literature to be associated with like fungal pathogens have these ceramide metabolic processes as part of their infection, as part of the interaction with the host. So that was one of the first moments we said, okay, we are going in a good direction, because we are maybe finding something that can tell us something about the pathogenic mechanism of this fungus. And then we found some DNA recombination metabolic processes in the exclusive. So yeah, we found a lot of interesting Go terms. We decided to check the variant and to do a variant analysis. We saw a lot of variation even in the core genome, which is also really unexpected. Like, well, the core genome was supposed to be very conserved across the species. However, we found a lot of variation. And we also found something really interesting, and is that most of the complex multi-chromosome arrangements in our genome is actually in intergenic areas of the pangenome. So what is happening inside this fungus, right? Just to some other stuff, in chromosome 9 and chromosome 8, we found really interesting stuff. We saw a density of 10 variants each 5 nodes in the accessory class. So okay, are we seeing some kind of pattern related to specialization, adaptation to new environments? And then in chromosome 8, we saw that the exclusive and the core class had the same variations, but in different densities. So are we seeing the specific insertions that can talk about something interesting going on in that pangenome? So yeah, as I said, I'm running out of time, but as I said, we have a lot of interesting Go terms, interesting protein domains as well that can be explored further to understand this fungus. And then we have this little side. We want to enter the machine learning group, right? And the AI group. So we want to use the promoter regions, pangenome class that we already have, and variant hotspots in order to train a convolutional network to see if we can predict the stage of the infection that a sample comes from. So yeah, I kind of already said the takeaways. I want to thank my lab, which is my family. And of course, I am more than willing to discuss this further. I will be here the rest of the day and the rest of the week. So thank you.
Thank you. Yes, question?
You said something. Okay, it's working. There's something interesting, namely that a large portion of the conserved regions are intergenic regions, right?
Yes.
And what is the percentage of intergenic regions in this genome?
That's a really good question. I don't really have the answer right now. I don't have that percentage. So maybe if you can come and see me tomorrow, if you'll be around, I can definitely bring that statistics for you.
Well, thank you, gladly. But is it as large, as high as in the human genome? No.
No, no,
No.
it's way smaller. Yes.
Yeah. Okay. Thank you.
Yeah.
Thank you. Other questions? Okay.
Now that you're doing all the running. So you have a very small core. And to me that says this organism is somehow able to and willing to take up foreign DNA and incorporate in the genome. Do you know anything about the mechanisms that might support that in this particular fungus?
So we've found some evidence that has kind of making us think that this fungus has been under some selective pressures and that it has passed through some events of horizontal gene transfer, and it has actually been reported in the literature. So we haven't actually proved that hypothesis yet, but it's part of the things we want to look into, because it's very interesting the way this fungus kind of adapts to different environments and a lot of different geographic ranges. So we think it's one of those events. Thank you.
Okay, one more question.
Thank you very much. When you found that when you built the pangenome and you have this kind of completely opposite trend of loads of specific and very few, and you did also the normal gene base pangenome, which I think is not the right word to use. One or the other is a pangenome. The other is just like a gene collection, I would say. But
Okay.
or at least a pangenome graph is not the same as a pangenome gene base. It's very different. And we are very used to use this kind of gene base pangenomes. So why is that so different?
So that's also one of the things we want to discover, because as I said, we actually built an ortholog-based pangenome before, and we found some stuff. Right now, we are just in the early stages of the graph construction. We've been working on this for 3 months, so we really we haven't really understood where this difference comes from. Like we
You
don't.
only have 5 genomes, right?
Yes, exactly.
So maybe most of those things that are specific, they are present in the other genomes that are not part of your
Yes.
in the gene-based thingy.
Yes, that could be one of the reasons. However, the difference we found between the core genome that we built before and the core genome that we have now it's surprisingly not that different. So that's when we thought, okay, why with 22 genomes we have some results, with 5 we have almost the same results? So that's something we are trying to look into right now, because it's really interesting. Thank you for your question.
Thank you, Isabella. And let's thank all the speakers of session again.